Hello, and welcome to this episode of our Active Motif webinar series. My name is Mason Brooks, and today we'll be hearing from my colleague, Dr. Michael Garbati, who is a technical support scientist here at Active Motif. He will be discussing ways that epigenetic tools and techniques can be applied to the study of specific disease states, and in this case, he's going to be discussing blood cancers.
Hi, I'm going to start by defining epigenetics. Epigenetics is really histone modifications and DNA methylation. So these aren't changes in the genetic bases, they're modifications to those bases. They change the shape of the DNA, the shape of the chromatin. The way that our DNA, our genomic DNA is organized is in chromosomes. You can see our DNA is wrapped around these proteins. These proteins are nucleosomes. Each nucleosome consists of 8 subunits called a histone, and these histones can be modified by post-translational modifications. Also, the DNA itself can be modified. It can be methylated on cytosine bases. So taken together, these histone modifications and the DNA methylation can change the shape of the chromatin within a chromosome and thereby change the function of that DNA. And we're going to dive a little deeper into what that shape looks like. When chromatin is opened up by some of these modifications, genes can be switched on. Genes and functional loci in the genomic DNA can be accessible. When the chromatin is condensed, these genes and functional loci like promoters and enhancers are inaccessible to DNA binding proteins, and those genes and functional elements can be turned off. So transcription impeded when the chromatin is condensed, and transcription is possible when the chromatin is open. And here I've shown by these acetylation groups that typically it's histone acetylation that is the functional modification of histones that opens up that chromatin, and DNA methylation is one of the modifications that closes that chromatin. So several of these modifications have been characterized. So histone H3 lysine 27 acetylation is one of these modifications that opens chromatin. On that same residue, if it's instead trimethylated instead of acetylated, that helps to close and repress the chromatin. Also, sometimes a methylation on a histone can be part of an active gene. For example, histone H4 trimethylation is a modification that is usually found at transcriptional start sites. A monomethylation of that residue is usually found at primed enhancers. So it's not a hard and fast rule that acetylation is always on and methylation is always off. In general, usually histones, when modified as to be acetylated, open up the shape of the chromatin, and when DNA is methylated, such as 5-methylcytosine, this represses and closes that chromatin. So we have several different ways to measure these things, whether they be the histone modifications or the DNA methylation. ATAC-seq, which we'll get into later, is a method of using transposase to sequence the accessible and open chromatin. So it physically uses a transposase to get into these open spaces where the chromatin is accessible to tag and sequence that DNA. ChIP-seq is a method in which we use an antibody that is specifically targeted towards a histone modification. MEDIP-seq is a method in which we use an antibody specifically against a methylation of DNA, either 5-methylcytosine or 5-hydroxymethylcytosine. And bisulfite sequencing does not use an antibody. Instead, we use an enzyme to convert all unmethylated cytosines to uracil, and then we perform next-gen sequencing on the DNA of interest. And when cytosine is converted to uracil, that means that cytosine base was not methylated. When it is protected and not converted to uracil, that signifies that base was methylated. So I'd like to introduce an example where some of these methods can be very useful in studying a disease state. So the disease I'd like to bring up is chronic lymphocytic leukemia, the most common leukemia in the Western world. However, its only genetic characterization is the mutational status of the Ig variable region. So when the Ig variable region is mutated, it's a less aggressive disease. When it's unmutated, it's a more aggressive disease. However, this disease presents itself in many different ways. There's many different phenotypes. So it can't be just the Ig variable region mutational status that defines the phenotype. There has to be some epigenetic deregulation that plays a role in this disease. So how would we go about studying a disease like this? So a workflow example as presented by Rendiero et al. In Nature Communications, their example is that they chose 88 different CLL patients, took blood from them and purified the lymphocytes. They performed ATAC-seq, which is the measurement of accessible chromatin, and also ChIP-seq against a few different histone modifications. They compared ChIP-seq to ATAC-seq, and they were able to form a map of the chromatin landscape in this disease. So let's take a closer look at ATAC-seq. ATAC-seq it stands for Assay for Transposase-Accessible Chromatin. So what we do is we use a transposase called TN5 that has indexes bound to it. These indexes are ligated onto the 5' and 3' ends of DNA that is accessible to the transposase. So the DNA gets cut, indexes get ligated, and then we can perform next-gen sequencing that DNA that was accessible to the transposase. So here's an example of what this ATAC-seq data might look like. Like I said before, Rendiero et al. Took 88 different patient samples and performed ATAC-seq. Here's just looking at the B lymphoma kinase gene. We can see that there are several different parts of this gene that are accessible chromatin in the CLL patients. Some of them are almost always open. Some of them are open in most of these patients, and some loci are open in half or some percentage of them. So not every patient performed exactly the same for this locus, but we have a general picture that parts of this gene tend to be open chromatin in this disease. Also, they correlated this with previously published data from ENCODE, showing that at the sites of open chromatin, we also see nearby a lot of H3 K27 acetylation, which, as I said before, is also a histone modification of open active transcribed chromatin. So looking at two other genes in CLL patients, we see that the ZNF667 locus and the ZBTB20 locus are differentially modified and differentially regulated in CLL versus the mutated CLL. And just as a reminder, the unmutated version of the disease is a more aggressive form of this disease. The mutated CLL is the less aggressive form. So looking at the ATAC-seq data for ZNF667, we see open chromatin at this promoter. We also see acetylation of the histone at this promoter, looking at the ChIP DNA, the ChIP-seq result. So what this shows us is that this gene is probably on. There is K4 monomethylation, that's the primed enhancer mark. That is on in the unmutated form. All these things are off in the mutated form, except for the K27 trimethylation. That seems to be on in the mutated form, and that is our mark signifying that chromatin is closed, that the gene is off. So these data correlate with each other, and for ZNF667, this shows us that that's a locus that's on for the UCLL and off the CLL. So conversely, when we look at the ZBTB20 locus, there's open chromatin, there's acetylated histones at this locus for the MCLL patients, whereas for the UCLL patients, this gene seems to be off, that chromatin is closed. There's lack of histone acetylation. There's less of the primed enhancer mark. There's a little bit more of the closed chromatin mark. So here we have an example of how we can take ATAC-seq data and histone mark ChIP-seq data to make a determination of an example of a gene that we can say is a marker of the aggressive form of the disease, and a gene that is a marker of the less aggressive form of the disease, even though these genes are not mutated. It's just that they're regulated differently in these two forms of the disease, and the way they are regulated is by how their loci are modified epigenetically. Another way that we can look at epigenetic regulation of genes in this disease is by looking at superenhancers. A superenhancer is a long-range, powerful enhancer cluster that is usually marked by H3K27. That's that activating mark of histones that opens up that DNA and lets transcription factors and DNA binding proteins move in and activate that DNA. We see in superenhancers that mark. We don't see DNA methylation. And these superenhancers can activate several different promoters at 50 kb to several megabase difference at those distances. So they're far away and they're multifunctional. And if they're dysregulated, they can affect many different genes. So this can also be, dysregulation of these superenhancers can also be something that explains CLL severity and whether the patient has a more or less aggressive form of the disease. So to study these, Ott et al. Chose to look at 23 different CLL patients. They looked at the lymphocytes that are CD19 and CD5 positive. They studied the ATAC-seq profile, so looking at the open chromatin. They did ChIP-seq for H3K27 acetylation, and took this data and had downstream applications of establishing transcription factor networks. They did CRISPR screens of those transcription factors that were highly expressed. And they also treated with some inhibitors, which we'll get to later in this talk. So looking at one locus that this group focused on, the BCL2 locus, we can see that in the CLL patients, the superenhancer that controls that locus had a lot of histone H3K27 acetylation. And the ATAC-seq data shows that locus was also open chromatin. So like I said before, usually K27 acetylation and open chromatin, that correlates. And so here we have a big region where there is a lot of open chromatin and a lot of K27 acetylation. This was just one of the superenhancers that this group identified. We also took a look at the promoters. Here we were looking at the BANK1 promoter and the CTLA promoter. And here we can look at a comparison between the CLL patients and NBC, which is normal blood cells, so healthy donors. And we can see an example of the BANK1 gene that is on and open in both the CLL patients and the healthy donors. However, the CTLA4 gene seems to be off in the healthy donors, but is highly expressed and on in the CLL patients, as shown by H3K27 acetylation and this open chromatin results from the ATAC-seq data. So looking at this gene CTLA4, we can assume that gene is highly expressed in CLL. So of course, they didn't just look at CTLA4. This group wanted to build a transcription factor network to look at all of the loci that are highly expressed or aberrantly open, aberrantly acetylated at K27 in this disease. And as we look at just these 4 examples, the top of the list has similar culprits. PAX5 seems to be up there in that it's aberrantly on, aberrantly open chromatin. The enhancers that control that gene are aberrantly acetylated at histone H3K27. So PAX5 is one of those loci that keeps popping up, as well as CXCR4. CD74 is also high in a lot of these samples. BCL2, CD5. So we have a list of enhancers. We have a list of genes that are turned on by some of the same enhancers that are aberrantly on and open. So how do we target these? So the authors also use CRISPR screens to target these culprit loci. And in doing so, they found that the transcription factors that were most effective in the CRISPR screens were commonly activated by these super enhancers that we discovered earlier. So some of the proteins that bind to and activate from these super enhancers are the BRD proteins. So one candidate inhibitor that could possibly work here is a BRD inhibitor, JQ1. And as we can see here, that inhibitor was especially effective in targeting these specific loci that were on our list of the culprit transcription factors in this disease. So to look at how this inhibitor worked in a mouse model, the treatment of JQ1 slowed the growth of xenograft tumors from CLL. So now we've been able to use epigenetic techniques to identify super enhancers that are aberrantly on in the disease, identify a group of transcription factors that are aberrantly on, and then target them not by targeting the transcription factor, but by targeting the super enhancers that aberrantly control the expression of that transcription factor. And so using an inhibitor against a super enhancer, we can suppress many different targets of that super enhancer. So lastly, I'm going to show another study on CLL where this group focused on chromatin states as defined by many different histone modifications. So to refresh, each one of these histone modifications has some different function. For example, H3K27 is an active promoter, a strong enhancer. H3K27 trimethylation is a mark of repressed chromatin. So if we take these together, Beekman et al. In Nature Medicine in 2018 decided to color code them so that we can get an average of the histone modification landscape. So if we look at the unmutated CLL, that once again is the more aggressive form of CLL, versus the mutated CLL versus normal blood cells from healthy tumors, we can see several loci that look completely different in the disease state versus the healthy state. The ATAC-seq data can also show that these hotspots of modification of histones that are activating in the disease state that aren't there in the healthy state also shows that these also correlates with the ATAC-seq data showing that there's open chromatin in the disease state that isn't there in the normal state. Layered on top of that is DNA methylation analysis. And the orange track is the disease state CLL, the gray track is normal B cells. And we see that at these same loci that have the aberrant histone modifications, they also have a loss of the DNA methylation. Also, we can see that the RNA-seq shows aberrantly on genes, an aberrantly on gene that is responding to these histone modifications and to the DNA methylation. So we can see a much better view of the chromatin landscape when we combine several histone modifications. We combine that with the survey of open chromatin, that's ATAC-seq, and we also look at DNA methylation and RNA-seq. We can get a complete look at the chromatin landscape in this disease. So let's look at one other locus. That's the TCF4 gene that they also studied here. Here we can see that upstream of that gene, there is a lot of activated chromatin in the mutated and unmutated forms of CLL versus in normal cells, that chromatin does not have these activating marks. It's not open chromatin as measured by ATAC-seq. It has a lot of DNA methylation, which is lost. That DNA methylation at that locus is lost in CLL. And that gene is highly expressed in CLL, whereas it's not as well expressed in normal B cells. So we can see now how histone modifications as measured by ChIP-seq, chromatin open and closed state as measured by ATAC-seq, DNA methylation as measured by MEDIP-seq, plus all taken together with RNA-seq, we can get a much better view of the chromatin landscape in CLL. And Active Motif has the kits and the services and the antibodies to perform all of these different techniques so that you can also get a good view of the chromatin landscape in CLL or any disease that you study that may have an epigenetic aspect to it. So in conclusion, combining epigenetic tools using blood samples can provide answers of why certain genes are up and down regulated in the disease state in the absence of some mutation. They can help establish a reference genome or a chromatin landscape for a disease, but we can also use this information to identify deregulated loci and maybe even identify some druggable targets for inhibition to help alleviate the phenotype of the disease. Thanks for your time. For the products and services that I mentioned a couple slides ago, please visit our website, activemotif.com. You can contact me directly at tech_service@activemotif if you have any questions about those products or services or any of the data presented here today.